Bismillahirrahmanirrahim. I am your instructor, Dr. Naila Khalid. Here I am going to discuss that new topic that is aliphatic nucleophilic substitution. But is this type of substitution is specifically SNI. In our previous lectures, we have discussed about SN1 and SN2. Now we are going to discuss this specific nucleophilic substitution reaction mechanism, which is known as SNI, internal nucleophilic substitution. It means that the internal nucleophile will attack, means that intramolecular attack of the nucleophile take place. So that is why this is special type of nucleophilic substitution, and definitely it is different from SN1 and SN2. So in this session, we will be able to have a clear concept of SNI reactions mechanism, internal nucleophilic substitution reactions. You will be able to identify these types of reaction mechanism and any factor which can affect this type of nucleophilic substitution will be discussed in this session. Definitely, we will discuss their reactions as well as their mechanism to give you a clear idea about SNI, how they are different from SN1 and SN2. So this is the part of your course. The chapter is aliphatic nucleophilic substitution and elimination, and this topic has been taken from your course. So here is the outline of our lecture. We will discuss what are different types of SNI reactions, what is their mechanism, how you can suggest their mechanism, and what is the stereochemistry of the SNI reactions. Especially here, you will see retention of configuration take place. No racemization, no inversion, but only retention of configuration take place. And there is an important role of addition of dioxane and pyridine. They will change the reaction mechanism from SNI to some other type of mechanism. How these affects the mechanistic pathway, dioxane and pyridine we will discuss. At the end, the summary, the major points. First of all, let me clear you SNI. Sometimes it is abbreviated like substitution nucleophilic internal. It is also said that this I is for intramolecular. Both are correct, but internal nucleophilic substitution. But basically, nucleophile is not the external one, but it is present inside the substrate molecule. Inside the substrate, if it is combined with some reagent, then it has an internal nucleophile. No external nucleophile. This internal nucleophile will attack. Definitely, this is an intramolecular attack from one side, and that is why you will obtain a different types of stereochemistry. On screen, you can see a very clear example of this type of SNI reactions. Here you can see that the SNI reaction is, the substrate is this alcohol. This alcohol, it is chiral in nature. So if it is chiral in nature and you will treat it with thionyl chloride, then this alkyl chloride is obtained. Now, what is the unique feature of this SNI? That is that the use of this thionyl chloride, this reagent is called thionyl chloride. Thionyl chloride, when attacked here, it will be attached, and then one of its chloride will act as a internal nucleophile, and you will get a nucleophilic substituted product in this way. But the unique feature of this SNI is that you will see retention of configuration rather than inversion, and no racemization take place. So this type of reaction is very important, and you will see that the use of this thionyl chloride have transformed this type of SN1 or SN2 into SNI, a nucleophile which attack from the internal side. Now come to the mechanism and rate of reaction. First of all, the rate of reaction. It follows the second order kinetics. Second order kinetics means what it means. What it means that your your rate of reaction is dependent on the substrate as well as this reagent. So it is dependent on these two species. So just like SN2, the rate of reaction is dependent on the two molecules, substrate and reagent. So that is why it follows second order kinetics, just like SN2. So when you study the mechanism, you will see one of the step is the rate determining step. So all the steps are not rate determining step. Only those steps, only that step is the which is very slow. It can be considered as rate determining step because whole of the time take place for to complete that reaction will be dependent upon this one. So this step is basically formation of alkyl chlorosulfide. This alkyl chlorosulfide is an intermediate. You will see its structure, its formation in the mechanism. So here is the rate of reaction of SNI, which is dependent upon the concentration of thionyl chloride and the substrate, just like SN2. But it is not SN2, it is SNI. Just like SN1, it passes through different steps, formation of some intermediate, but it is not SN1 or SN2. It is special type of nucleophilic substitution which is known as SNI. So here on the screen, you can see the clear mechanism of this type of alcohol. If this is alcohol and these are different alkyl groups, this is a chiral one. How it attacks on thionyl chloride, this is the structure of thionyl chloride. Oxygen with its lone pair attack on sulfur, and this will be broken down, and a new bond is formed between oxygen and sulfur. And in this way, this hydrogen is abstracted by this chloride because this electron density comes here. Chloride is removed from this surface, and this chloride will abstract this proton, forming a byproduct HCl. So H from here and Cl from here. HCl is formed, and this electron density comes here. So that is why you will get this type of intermediate. It is known as alkyl chlorosulfide. The formation of this intermediate, this step, is the slow step, and it is also known as rate determining step. And then what will happen here? this chloride, which is present inside this molecule, it is acting as an internal nucleophile. Along with this electron pair, it will attack on this electrophilic carbon. This electron density comes here, and you will get another stable molecule, which is sulfur dioxide. So this is also the byproduct. Sulfur dioxide is also the byproduct, and it leaves the surface. At the end, as this nucleophile has attacked from here, from the same side to which this leaving group was attached, so this is also leads to a retention of configuration. Any, it means that the configuration of the substrate and configuration of the products both are same. It has not been changed. So you will get exclusively this type of isomer with retention of configuration. So I will repeat over here that in this type of reaction, you will get the two byproducts, sulfur dioxide and HCl. So the rate of reaction, which is dependent upon both thionyl chloride and substrate. So in this context, SNI reactions resemble SN2. Rate of reaction is the same as that of SN2. However, in SN2, the special feature was observed that the attack of the nucleophile was from the back side, and we have obtained inversion of configuration in case of SN2. But in case of SNI, you will observe no inversion of configuration, no racemization, but only retention of configuration, as we have discussed on slide number 7 previously. So, retention of configuration is the basic feature of SNI reaction, and it is the evidence that how the nucleophile is attacking from the same side and internal attack take place. Here is another example of a secondary alcohol, which is also chiral in nature, 1-phenyl ethanol. And this, if it is treated with thionyl chloride, we will get 1-phenyl ethyl chloride. The configuration of the isomer, if it is R, then it is also retained. Here it will be also R. And if it is S, then it is also S. It means that the configuration of the substrate will be same as the configuration of the products. And the byproducts are sulfur dioxide and HCl. So here is the mechanism of that 1-phenyl ethanol. This is 1-phenyl ethanol. How this electron attack on this sulfur chloride is removed, and you will get this type of intermediate transition state like this. Actually chloride is removed, and that chloride will extract this proton, electron density come here. So this is chlorosulfide intermediate, and it is the rate determining step, that chlorosulfide formation. Here this chlorosulfide is decomposed to give you the sulfur dioxide. How this whole chloride with electron pair will act as a nucleophile, will attack on this electrophilic center. Electron density come here, sulfur dioxide is removed, and you will get this exclusive product, exclusive isomer with retention of configuration. So 1-phenyl ethyl chloride is our product, and the same configuration has been achieved. So here on your screen, you can see another example. This type of alcohol, it is secondary in nature. This is This is special type of substrate which contain this carboxylic acid group. If the two carboxylic acid group are present, then this is called dioic acid. And this is 2-hydroxy 1,4-butane dioic acid. So this hydroxy get substituted. This is substituted by, if it is treated with thionyl chloride, then this type of attack, and then this take place, and then chloride is removed, and that chloride will attack from the same side, and you will get this chloride will obstruct this hydrogen, electron density come here. This is the uh, this is the chlorosulfide group, chlorosulfide intermediate, chlorosulfide intermediate. Here it is the rate determining step. Chlorosulfide group is then decomposed. How this chloride will act as an internal nucleophile, will attack on this electrophilic carbon, and then this electron density comes here, and sulfur dioxide is removed in this pathway. So you will see that in this internal nucleophile, this is some of the explanation that there is possibility that this chloride attack from here and this comes here, sulfur dioxide is formed. And there is also a concept that before this during the decomposition of chlorosulfide, the intimate ion pair is formed, and this intimate ion pair is formed in this way, and this nucleophile will attack on this carbon, and you will get nucleophilic substituted product after the loss of carbon as sulfur dioxide. So this mechanism shows that how the intimate ion pair is formed by the decomposition of chlorosulfide. What is the stereochemistry of SNI reactions? As I have mentioned before, that this type of reactions involves only retention of configuration. So you will get enantiomerically pure product without racemization. So it resembles SN2 in terms of rate of reaction. It resembles SN1 in terms of its formation of intermediate and stepwise mechanism, but it is different from both SN1 and SN2 because like SN1, there is no racemization. You will get enantiomerically pure product. And SN2, there is inversion of configuration, but in case of SNI, you will observe retention of configuration only. So SNI reactions are affected by the addition of some electron pair containing reagents like pyridine, dioxane. So here in case if you are adding pyridine to any SNI reaction, there is possibility then the stereochemistry of the product is changed. And you will observe here inversion rather than inversion rather than retention of configuration. Just like SN2, if you add pyridine, now the mechanism is no more SNI, but it is SN2. No retention take place, only inversion take place. Now the question arises, how this inversion take place? This can be answered by writing the or suggesting the mechanism. Here is again the same example, which if it is treated with thionyl chloride, then chlorosulfide has been formed. I have discussed in previous slide too that, but in absence of pyridine, this chlorosulfide is going to decompose to give you the product with retention of configuration. But if this pyridine is added, this pyridine has electron pair, and this electron pair is easily available because it is not the part of aromatic ring. This will attack on sulfur and this is opened. And when it's come back, this chloride is removed. And the pyridine has blocked this side in the form of pyridinium ion. So that is why the chloride which has left the surface over here, this will attack from the back side. And from the back side, and this electron density comes here. Sulfur dioxide is formed, this electron comes here. Pyridine is obtained at the end, acting as a catalyst, blocking front side, making nucleophile to attack from the back side. So that is why instead of retention of configuration, you will obtain inversion of configuration if you have added pyridine. What is the impact of dioxane? Dioxane is also, this is the structure of dioxane. Dioxane has also electron pair on oxygen, both oxygen. One of the oxygen from here, it will attack this dioxane. This will attack on this electrophilic carbon. Electron density comes here because this is the same step in each case. As second chiral alcohol, when it is treated with thionyl, you will get chlorosulfide intermediate. And when you add dioxane, it will attack on this electrophilic carbon. Electron density comes here, and you will get sulfur dioxide, and dioxane has blocked this side. So the chloride will have possibility to attack from this side. And that is why as dioxane has blocked the back side, the chloride will attack from the front side. So you will get retention of configuration because in this case dioxane has blocked one of the side. In case of pyridine, it will attack on this sulfur. So that is why it will block the front side. Chloride will attack from the back side. You will get inversion of configuration. So using pyridine or dioxane like reagents, you will get with pyridine, you will get this type of inversion, retention, rather than retention of configuration, you will observe inversion of configuration. However, this chlorosulfide, if it is decomposed and in such a way that intimate ion pair is formed, then the chloride will attack from the front side, and due to internal nucleophilic attack, you will see retention of configuration. So here is some assignment. I will explain this. The question number 1 is explain the stereochemical outcome when a chiral alcohol reacts with thionyl chloride in the absence of pyridine. You have to take any chiral alcohol and you have to write the equation with this chiral alcohol as a substrate, thionyl chloride as a reagent, and the product is a nucleophilic substituted product. That is, in the absence of pyridine, it will give you which one? Retention of with the product with retention of configuration. So the second question is that why does the addition of pyridine change the mechanism from SNI to SN2? इसके लिए आपको मैकेनिज्म जो है वो बाकायदा ड्रॉ करना है with a specific chiral alcohol. So you can explain this reason that addition of pyridine has changed the mechanism from SNI to SN2. So discuss the role of ion pair formation. We have discussed that how ion pair is formed in internal nucleophilic substitution reaction. So you have to discuss that what is the role? The important role is what? It is the configuration retention. Retention of configuration has been observed, and this is due to the formation of ion pair formation, and which blocks the from the front side, the internal nucleophile will attack. So the last question is that you have to compare the kinetics, means rate of reaction, kinetics and stereochemistry of SNI with SN1 and SNI with SN2. So this is the end of this session. I hope you have learned some basic features about SNI reactions. So this is the end of this lecture.